Hello everyone, I'm Tiede. I'm a master's student in the Department of Kinesiology at IUPUI, and today I'll be talking about early muscle gene expression changes with eccentric exercise uniquely associated with peak strength loss. Currently, we know that unaccustomed exercise, particularly those emphasizing eccentric contraction, will result in muscle soreness that persists for days following exercise. This phenomena is referred to as DOMS, or delayed onset muscle soreness. In addition to DOMS after eccentric exercise, we often see disruptions in sarcomere structure, such as Z-disc streaming or sarcomeres out of register, as you can see in the picture to your right. These temporary changes in structure can also cause disruption in the excitation-contraction coupling process, which is thought to be a secondary cause of strength loss following eccentric exercise. We often see a high degree of variance in the severity of soreness observed in individuals following eccentric exercise, what you might think of as high or low responders, which is what we want to investigate further. The mechanisms behind this variation are poorly understood. Thus, the purpose of this study is to use global gene expression profiling to investigate transcriptional muscle RNA changes related to peak strength loss to identify important biological pathways. In a previously published study, a cohort of 35 untrained healthy male subjects participated in eccentric exercise conducted on a Biodex isokinetic dynamometer, similar to what you see in the picture here. Subjects performed 10 sets of 10 eccentric knee extensions at an angular velocity of 30 degrees per second. In between each repetition, participants rested for 10 seconds, and in between each set, they rested for a minute. Strength was assessed 1 day prior to exercise, immediately after exercise, and then every 24 hours after exercise for 5 consecutive days. Strength testing consisted of three 3-second maximal voluntary contractions of the knee extensors on the Biodex. Following the exercise bout, a muscle biopsy was obtained from the vastus lateralis of both the exercise and non-exercise leg, which was used as a control. RNA was isolated using the TRIzol method, and Agilent Whole Genome Microarrays were used to form transcriptome profiles. Using Pearson correlation coefficient, peak strength loss was compared to changes in gene expression in the exercise leg relative to the control leg. Then using IPA software, biological functions of correlated gene sets were analyzed. We found that strength loss averaged 50.7 plus or minus 20.1% with a range of 9.7% to 96%. As you can see from the wide range, individual response varied greatly. We detected 2,333 genes relating eccentric exercise to peak strength loss. IPA analysis revealed 37 canonical pathways associated with peak strength loss. Included in these pathways were glutamate receptor signaling, GABA receptor signaling, and cyclic AMP mediated signaling pathways. In our analysis, we were able to identify biological pathways that relate to severity of muscle dysfunction following exercise, including neurotransmitter pathways such as glutamate and GABA receptor signaling, as well as key cell signaling pathways such as cyclic AMP. More insight into the involvement of these pathways in strength loss could lead to possible therapeutics that can prevent high strength losses following exercise in those prone to muscle dysfunction. Upon further research, there could also be possible implications into targeting these pathways to optimize the use of eccentric exercise to improve function without damage in athletes. Strength testing was performed by experienced technicians, and subjects were provided verbal encouragement throughout. Despite this, it is possible that subjects may not have expressed their full ability to contract due to other factors such as psychological state. In our study, we were able to identify key pathways associated with strength loss, but we did not test direct causal relationships, something that we can follow up with on future studies using functional in vitro models. Thank you to everyone who listened for their time. Please feel free to leave a comment with any questions you may have.